swear? You were sent by the agency too, huh? Looks like we're both on the same mission to figure out what's causing the ruckus in Proteenia. I've already uncovered a few clues that might lead us to the source of the trouble. Here, take a look with your magnifying glasses. They'll help you see the hidden details. Let's crack this mystery wide open. Hey, you found a primary structure. Remember this amino acid sequence. This might be crucial for your journey ahead as it holds the secrets to how the culprit functions. You seem to have found how it is folded. This is its secondary structure. This evil thing arranges itself very carefully, with its sneaky beta twist and deceptive alpha helices. It employs disulfide bridges as its dark chains, locking its form into a treacherous cyclic structure. This precise configuration is what gives it its menacing power. If it were to misalign or misfold, its malevolent plans would unravel, and it would lose its edge. It was nice meeting you. I hope you pick up the things that I've said, as it will be very useful until the end of this game. Goodbye for now.
Welcome, traveler. Stocking up for your journey, I see. Excellent choice. Say, are you one of the adventurers heading to Protonia? If so, I got just the thing. Magic vision glasses. Trust me, these will help you reveal secrets hidden within this ancient scroll. I see. It seems like you have found more hints on who the culprit might be. Shown on the scroll are the tertiary and quaternary structures of the protein you're looking for. In the tertiary structure, it can be seen that there is a 1 to 7 interchain disulfide ring bridge held by covalent bonding between the two cysteine residues. It also contains a hydrophobic core through the linkages of nonpolar amino acids of choline, phenylalanine, valine, leucine, isoleucine, and methionine that also contributes to its stability as well as its globular structure. Furthermore, it is also relatively compact, very demure, very cutesy. In its 3D structure, the disulfide bonded cysteine residues forming a ring can be seen on the N-terminal side of the image, followed by the alpha helix region, then its amidated residue, which means that instead of a carboxylic group, an amide group is found at the C-terminal. The one we're looking for does not seem to have any quaternary structures, mainly because of its size and function. It only contains 32 amino acids and is stable on its own. Additionally, this hormone binds to its receptor at the osteoclasts, renal, and neural cells with ease. Bottom line, its primary, secondary, and tertiary structures are enough to fulfill its biological role. However, there have been studies where it forms dimers or oligomers under certain conditions only, because those aren't really its predominant forms. So, this is the troublemaker? But something's off. A lot of the villagers here share similar traits. For all we know, it could be amylin or CT gene related peptide, pulling the strings instead. I think it's time to call for backup from the wizard and the princess.
It was nice meeting you. I hope you picked up the things I've said since it will be very useful till the very end of this game. Goodbye for now.
Alright, so let's dig in and see what we've got. Time to crack open our spellbooks. They might just hold the key to solving this puzzle. So for the function and significance, calcitonin is a hormone primarily produced by the parafollicular cells, also known as the C cells of the thyroid gland in humans. Its primary functions include, first, the regulation of calcium levels. Calcitonin helps to lower blood calcium levels when they are too high. So the second is inhibition of bone resorption. By reducing the activity of osteoclasts, calcitonin decreases the rate at which bone is resorbed, helping to maintain bone density and strength. Third is the promotion of calcium excretion. Calcitonin increases the excretion of calcium and phosphate by the kidneys, further aiding in the reduction of blood calcium levels. Last but not least is the regulation of phosphate levels. In addition to calcium, calcitonin also helps regulate phosphate levels in the blood, often acting in concert with other hormones like parathyroid hormone or the PTH. While calcitonin's role in calcium regulation is important, its effects are generally considered to be less significant compared to those of PTH and vitamin D in maintaining overall calcium homeostasis. Hmm, I have heard that this calcitonin has a twin. Let's find out who it is so we won't be confused. Calcitonin or CT and calcitonin gene related peptide CGRP are both peptides produced from the same gene but are distinguished by their structure, function, and tissue of origin. So here's how they differ. So, focusing on the function, calcitonin or CT is involved in calcium and phosphate metabolism, and it lowers blood calcium levels by inhibiting osteoclast activity and increasing renal calcium excretion. So, for CGRP, primarily functions as a potent vasodilator, playing a key role in cardiovascular regulation, and it is involved in the transmission of pain and migraine pathophysiology. CGRP does not directly influence calcium metabolism like calcitonin.
Welcome, brave adventurer. I'm the Princess of Protenia. It seems like you have already discovered much about the mysterious disturbances plaguing your land. Your journey has taken you through the depths of protein structure, and now the final challenge lies ahead. But before you face it, you must understand the true essence of calcitonin, not just for your land, but for the whole wide world. Before we delve into the applications of calcitonin, it is important to understand how it differs from a related peptide called CGRP. Calcitonin regulates calcium levels produced in the thyroid, and it acts on bones and kidney cells. CGRP, on the other hand, is involved in vasodilation and pain signaling produced in neurons. So, CGRP acts on blood vessels and neurons. In summary, while calcitonin and CGRP originate from the same gene, they are distinct peptides with different physiological roles, target tissues, and receptors. Now, let's explore the significance of calcitonin in medicine. Calcitonin is not just any protein. It's more than just a regulator of calcium and phosphate levels. In the medical field, it is used to treat conditions such as osteoporosis and Paget's disease. Its ability to inhibit bone resorption makes it a powerful tool in maintaining bone density and health. Calcitonin is also used to treat hypercalcemia, where too much calcium is present in the blood. It is given via injection under the skin or into the muscle. Beyond the medical realm, calcitonin is also invaluable in biotechnology and pharmaceutical industries. It is used in the production of synthetic peptides as its analogs are being explored for new therapeutic uses, and it serves as a model for developing other peptide-based drugs, specifically those affecting bone health and calcium metabolism. The balance it maintains in Protenia mirrors its importance in these fields. Now, armed with this knowledge, you can see how the chaos in Protenia can cause the potential disruptions in these vital applications. The disturbances you've encountered threaten not just our beloved land, but the very balance of these crucial processes. It's time to make things right. With your newfound understanding, use this key to restore the balance. Track down the source of disruption and stabilize the calcitonin levels in Protenia. Only then will peace be restored and our land can thrive again.